PowerPoint reference the sheep data that we collected as well. And so for those who have interest in sheep as well as goats, that might be interesting. It also is kind of interesting because while there are a lot of similarities between the two species and how we manage them, the results were a little bit different, and certainly haven't looked far enough into the difference between the results by species to know whether the differences are statistically significant, but at least in a descriptive sense, they're interesting. I do cover St. Lawrence, Jefferson, Franklin, and Clinton counties here in New York. And both the sheep and goat version of this research project included those 4 counties, along with Lewis County and Essex County. And so I want to do a big thank you at the beginning to Doctors Ashley and Ellis, who are my co-workers for Lewis and Essex County, respectively, who did the sampling for the herds and flocks in those counties to help get this project accomplished. So one of the first things that we wanted to do was to kind of get a better idea of the diversity of the goat herds within the region. And we did the same thing for the sheep flocks in 2019, just to kind of better describe the population that we were sampling. And so Betsy and I kind of put together a survey, and Betsy sent it out on her listserv, and I sent it out to the farms that I had that participated in the New York State Sheep and Goat Health Assurance Program, which is a program that New York State has developed to help sheep and goat farms with managing and controlling and preventing Johne's disease already. And we also had some veterinarians that have a small ruminant interest that sent our survey out to their clients as well. And through that survey, we asked a bunch of management questions. We asked about size of their herds and flocks, years in the industry. We asked some questions about record keeping and parasite control and what other species they had on their farms and what they used their livestock for. But then we also, one of the most important aspects of this was then to also say, this is a project that we're doing. Are you interested in participating? And so certainly lots of people filled out the survey and said that they did not want to participate in the sampling component of our project, but many people did, and we appreciated those that did. And then we used that information about whether or not they were interested in participating and also their size to kind of determine who we were going to include in the sampling component of the project. How we used the information and what cutoffs we used for who we included varied a little bit between the sheep and the goats. For the goats, we included anyone who had 10 or more adult animals over the age of 2 who were willing to participate. And then we did include a handful of smaller herds that we were already going to be visiting because of their participation in the NICE-GAP program. And so it was kind of, if I was already there, I'd already made the drive out there, and we're spending the time doing the same kind of additional data collection and education that we would with the project. And they only had a small number of animals. It was really easy to just go get those samples while we were there so that we could get to the sample size for the total population that we wanted. So you'll see a little bit of a difference between sheep and goats there. For sheep, we said that they had to have a minimum of 15 adult animals that we could sample, and every flock that we included met that minimum number. And then, so the farms that we included, we did some, we had discussions about Johne's disease, and if they had had any history of yonis or anything that, might be concerning for yonis. And then we went out and we took blood and manure from animals that were over 2 years of age. If they had more than 20 animals, we only sampled 20, and we tried to sample them in as random of a approach as possible. So if they had 40 animals, we would try to sample every other animal. If they had 60 animals, we'd try to sample one and then skip two, and sample one and skip two, so that we wouldn't just be sampling the 20 friendliest animals. And if they had fewer than 20 animals, we would sample everyone over 2 years of age and include them. So I guess, taking a step back, what is Johne's disease? I mean, why are we spending all this time and effort to test animals for it? But Johne's disease is kind of like, the closest thing in human medicine that we can kind of make it analogous to is Crohn's disease. And that Crohn's disease also causes a thickening of the GI tract, which kind of makes it difficult to absorb nutrition from the food we eat. Certainly it is different in the causative agent. Crohn's disease is considered immune mediated, and Johne's disease is an infection that's caused by Mycobacterium avium subspecies paratuberculosis. And you'll see in some of my slides, I abbreviate that to MAP, which is a little bit shorter than saying Johne's disease, but more often than not, when I'm talking, I'm just going to call it Johne's disease. It is a fecal oral transmission. So the way that animals get infected is that you will have adult animals in the flock that are infected, and they are shedding the organism in their manure. And we all know that little baby goat kids and little baby lambs like to put their mouths all over everything. And so when they're putting their mouths on different things, if that stuff is contaminated with manure that contains the Johne's organism, they're going to ingest some of that organism. And it is very difficult to know exactly, how young is young enough to be, really susceptible to infection, and what amount of organism is needed to develop for them to develop infection later in life. It's, we don't know that and exactly. And also, some of it could be if they're exposed to a low level of it over, a couple weeks or a couple months versus getting one large dose of it at one time in their young lives. It's hard to quantify to know exactly what dose of it any animal gets versus will they or won't they develop disease later in life. And that's the other hard thing about this disease, is that animals get exposed when they're young, and then they may not show any clinical signs of disease until they're much older. They might be 7, 8, 10. I know that some of these herds and flocks have animals that are, that do have the longevity to be around otherwise for years within a herd or a flock. And some of those animals may also never actually develop clinical signs. And even if they don't develop clinical signs, they may or may not then begin to shed the organism themselves later in life and continue to perpetuate the infection. The other thing that makes this disease really difficult is that the symptoms are pretty non-specific. And so as veterinarians, it can be really hard for us, or even as producers, it can be hard for you to look at an animal that's sick and be suspicious of Johne's disease. It's a little frustrating because Johne's disease, when it manifests in cattle, has a very specific presentation. Those cows continue to eat. They're bright-eyed and kind of happily looking at you and eating, and they have terrible diarrhea, and they lose body condition very quickly. And so it makes sense because they're able to eat, they still have an appetite, but they're just not absorbing the nutrition out of their digestive tract. So they're pooping it all out, and their body is using the tissue and energy that's stored in muscle and fat because it's not absorbing any out of the diet. But sheep and goats are different. They kind of have this they can wax and wane. They can be not feeling well and not eating, and then they can rebound. They sometimes will have diarrhea, but sometimes they don't. They So it's very non-specific and can be really hard to pinpoint and say that there's a problem. The other hard thing is that that kind of waxing and waning, losing condition, not feeling great, can also go hand in hand with what you might see for parasites. And so Betsy and I did do a little bit of questioning in our survey about parasite control because it is a disease that can be difficult to differentiate from Johne's disease without additional testing. And certainly you might not just have one or the other. There are animals that certainly might also have both. So you might say, this animal looks this way and their FOMACHA score leads me to believe that it's parasites, but they're not getting better. So when I deworm them, so maybe they have resistant parasites, or maybe they had parasites and have, and that's resulting or in the FOMACHA scores, but maybe they also have Johne's and that's why they're not getting better. And sheep and goats are very interesting from a Johne's disease perspective because the way that we manage these animals makes it a lot harder to control the disease. And I'm going to kind of again refer to cattle and more specifically to dairy cattle, in that the dairy industry has made tremendous strides in controlling Johne's disease in that population. And they have done a great job through two tracks. And the first of which is management. We do make a very concerted effort in the dairy industry to remove young animals from more mature animals so that we do not expose those young animals to the pathogens and the diseases that the adult animals have. And also the dairy industry has spent a lot of money testing and culling animals with Johne's disease to manage their animals, that will manage their animals more similarly to how we use dairy cattle. But that certainly is not the norm in the small ruminant world. But we utilize pasture much more intensively. And so we're having animals eat the same, from the same area that they are defecating. And so that makes it difficult to stop or reduce that exposure of your food to fecal pathogens like Johne's. We also, there are a larger number of very small herds or flocks. And so we have more movement of animals between those groups, very commonly of bucks or rams for breeding, so that not everyone has to always have their own males on the property all the time. And so then you are potentially spreading disease through that movement. And we also raise our young stock with adults. And so it makes it really difficult to prevent the young from being exposed to manure from older adults. And then the last part is testing cost and logistics. And we'll go into this more, but to kind of put it in perspective, the testing that Betsy and I did for this project, if we hadn't gotten a discount for the research project, it would have been $85 an animal to do the 3 tests that we did. And certainly we're not advocating that everyone tests their animals using every test on the market. But there are each test kind of has flaws and benefits. And from a research perspective, doing all 3 kind of helped make it as robust of a picture as possible. And so because of those flaws, spending money on a single test may also not give you as much usable information to help make decisions. And we'll go into that further. So we kind of talked about Johne's disease being a fecal oral transmission, but this kind of illustrates that more. Um, an animal is exposed to contaminated manure and ingests it, and then it may take, years for them to develop any signs of disease. They certainly might shed organism into their feces before you see any clinical signs of disease. And that organism has infected and kind of taken up residence in the digestive tract, not just the intestines itself, but also the lymph nodes along the digestive tract, and even can get into the white blood cells. And so it can also be shed into colostrum and milk, which is another way that young stock can ingest it. And then even there's, a small percentage of animals that might even become infected in utero through a little bit of blood transfer or movement of bacteria. There's more than one strain of Johne's disease, and so sheep have the potential to have their own sheep S strain, and that strain is more difficult to culture and is more slow to grow. I did also find some research that shows that clinical animals are more likely to have a positive result on a test, which makes sense. With all of these tests, we are going to get the best answer when animals have actual clinical infection, as opposed to when we're testing animals that otherwise appear healthy. Goats are more likely to be infected with the cattle strain, and fortunately, those strains are more easily cultured. And you can actually, when I get to the slide where we look at the results, you'll actually be able to see that we were far more successful in our culture tests for goats than we were for sheep. So we really were able to see that in our two sets of results. So we did already kind of talk about the clinical signs. You might not see them for years, but we're really looking for weight loss. Some animals will have diarrhea. Some animals will get this edema or, almost like a fluid-like accumulation on their chin, which we call bottle jaw. And that's because they're not able to absorb protein as well. And so their blood can't keep fluid in the bloodstream, and that fluid kind of starts to migrate into the tissues. And so they get this kind of edema under their chin. Eventually, that disease will lead to emaciation and death because they just can't absorb enough nutrition out of what they're eating, if they're eating. But the biggest concern is the animals that are not symptomatic, or the animals that are able to spread disease before they become symptomatic. They're like the silent shedders and spreaders. Oh, so how do we combat Johne's disease? And this kind of and prevent bringing it into our farms. And certainly if you're on the market for new animals, buying them from people who test their animals regularly for Johne's disease is going to give you some security, at least at a minimum, testing a particular animal or animals before you purchase them and move them onto your property. While may not be perfect, is better than nothing, and at least maybe you catch something. I'm always a big advocate for quarantining animals upon arrival if they're not previously tested, so that you can test them. It's certainly not a guarantee that you're going to see signs of disease develop while they're in quarantine, but at least they're going through a stress period, and they may develop signs of disease or signs of other diseases that you then can pick up before you bring them into, in with the rest of the herd or flock. Management. So the management is hard depending on how you're raising animals. Really, the goal is with yonis is to limit exposure of young stock to adult manure. And that's really hard when we raise babies on dams, because those babies are going to be with adults. And that's, without, there's no other kind of way around that unless you're going to go through the effort of milking and raising bottle babies, which is time consuming and kind of a pain. But certainly that is a way to manage it. You can certainly do routine testing for adults, or at least testing animals that show any signs that could be Johne's disease. And then that way, if you do have an animal that you identify with it, you are aware that it's in your herd. If you do routine testing of adults and you find animals that have test results that are suspicious, you could, possible you could separate them and kind of manage them as two separate groups, those that are suspects or known positives, and then those that are negative. And then that way, you might choose to manage the babies from the ones that come out of the suspects more intensively to reduce the chance of exposing those to the adult manure that might be a higher risk. Certainly easier to do is to minimize overall fecal contamination of things animals are going to put in their mouths. So making sure that feed can't be contaminated with manure, water troughs can't be contaminated or are frequently cleaned. And then if you have animals that come up positive on a test, removing them, and so that you aren't, having additional time with known positive animals, putting your young stock at risk. Financial impact of Johne's disease. So I will say there's more research out there on the financial impact of Johne's in sheep than there are in goats. I did find a couple papers that were referenced for goats, but several of them were in languages that I don't speak or read. And so I didn't want to reference a paper that I couldn't actually read for myself. But there is a paper showing that for goats, there does appear to be a decrease in fertility for animals that are subclinically infected, but that decrease is only seen once those animals reach their 4th lactation. So they do have to be on the older side to start to see that. And then another paper that looked at profit efficiency in regards to Johne's disease. And they did see that profit efficiency goes down in a pretty linear fashion compared to the amount of Johne's disease that was present in the herd. And so you can see that with their graph on the right. And they found that that was true because of a decrease in milk income, an increase in veterinary cost, and an increase in feed costs. And so that makes sense. If your animals aren't able to digest nutrition as well, they're not going to be converting feed into energy as efficiently. And so you'll have to feed them more to get the same productivity out of them. So we had our survey, and this is kind of the distribution of herds that responded to us. We did have 47 herds that responded, and between all of those herds, we had just shy of 2,000 goats. And the spread around the North Country was pretty interesting. St. Lawrence County and Clinton definitely had the most that responded to us. The hard thing to know is how true this truly represents the true population in the area. I mean, certainly, I'm sure this is true for Betsy as well, but I drive around the North Country a lot, and I drive places and I see goats on the side of the road, and I think to myself, that's not a place I know about. I know that they didn't fill out the survey. I wonder what the survey would be like if truly, I could have spent the whole year stopping at every place I passed and asking every herd to respond and answer our questions. Our distribution certainly might be skewed based off of the veterinarians that worked more with us. We do have several veterinarians in St. Lawrence County and one in Clinton County that have very more intense practices that focus on small ruminants. We do have one veterinarian in Lewis who does a lot with small ruminants. But this was pretty interesting. I found it interesting that kind of 2 counties seemed to be a lot of it, but then Jefferson had 8, and that was over 600 goats by itself. But as the distribution by size was also very interesting. So by far the majority of people had 10 or fewer goats on their farm. And we had very few large herds, only 2 that were over 100 and 1 that was in the 60 to 70 animal range. And that was, when we think back to the sheep version of this, we had far more, 50 and over sheep flocks. So goats seem to be a small herd dominated group. We asked people what they used I forgot to change that from sheep to goats. God, I've done this twice already and I didn't catch that. We asked people what they use their goats for. And we asked primary purpose, and then on the inside it's secondary purpose. So 41% of people for their primary purpose use goats for milking, and 17% use that for their secondary purpose. The next 2 common are meat goats, which was 23% as a primary, and then 9% use that as their secondary purpose. Showing was big, and so 19 for primary and 23% for secondary for showing. And then breeding stock, which, to me is kind of goes hand in hand with showing, because lots of people who show also sell breeding stock. 15% as a primary purpose, but 43% use breeding stock as their secondary purpose. So that was really big. We did have 2% have pet goats as their primary purpose. And then 2% also use their goats for fiber as a secondary purpose. Other species. So we asked what else was on the farm, kind of because of concerns about strains of yonis, and the fact that other species beyond sheep and goats can have Johne's disease. It would have been really interesting if we had been able to do strain typing of the samples that we took, but that was beyond the scope of our project, maybe a future project. But 40% of people had cows, a little bit more than that had horses, a little bit over 30%, so about a third of people also had sheep. Lots of people had poultry, a handful had camelids, some had guardian dogs, and then about 25% had pigs. And we have been seeing the number of pigs on farms increasing since 2020 as well. By far, not as many breeds represented in the North Country compared to what we saw for sheep. And our population was very much dominated by alpine boar and Nigerian dwarf goats, but we had a smattering of other things as well. We asked kind of questions about how they housed animals around the time of kidding, and a big part of that is when they're kidding, those are the young stock. Those are going to be the animals that are at the highest risk of being susceptible to disease for ingesting manure and being around adults. And so 15% kidded on pasture, 36% in group pens, 45% in individual pens. So at least then you're minimizing your exposure of your kids to just the dam and not all the adults. And then a few people didn't answer that. And then we also looked at kidding times, which was different than the distribution we saw in sheep. By far, pretty much everyone's trying to kid out in this early to mid-spring time period. where the weather's probably going to be a little bit better than it would be earlier in the spring. And then they can kind of take more advantage of pasture later in the summer. I also asked about milk consumption. My background before starting in this position was with quality milk production services. So I spent a lot of time thinking about milk and milk consumption and raw milk. And so I'm always curious about what people do with the milk. And 59% of our responders milk their goats. Of that, 13% always pasteurize the milk that they milk, and 19% sometimes pasteurize. 75% of people made soft cheeses, 35% hard cheeses, 71% used the fluid milk in their homes, 61% would feed fluid milk to other animals, and 61% also used milk for non-food items. Record keeping in the goat community was far better than what we saw with the sheep. So most people are keeping kidding records, even more are keeping some kind of health records, and then about 2/3 keep some sort of pedigree information. And so record keeping in this group was really good. We also asked about different management tools for parasite control. Lots of people are doing FOMACHA scoring in some context, either regularly or when they see other clinical signs. Getting about 1/3 of people who are doing some fecal egg counts. Some are doing pasture rotation or multi-species grazing. A handful are doing some selective breeding. Lots more are keeping animals on dry lots so they don't have to worry about parasite control. Most people are only deworming with some other level of information. And really, we're at the point where only a small population is blanket deworming, which is excellent. When we first put together the sheep version of this project, our original goal was going to be to target flocks that had a higher risk for Johne's disease. So we tried to ask some questions to kind of feel out if Johne's disease was a problem in the herder flock. We weren't able to actually use that as a component of our selection and inclusion criteria, because if we had done that, we would not have had enough samples. But it's still interesting information. We asked people if they had had any animals die in the last year, and about 50% had. A slightly smaller percent had had a death from an unknown cause. There were certainly some farms included that knew they had Johne's in their population. So the orange percentage of the bar is yes to any of these questions. About 1/3 had seen clinical signs consistent with Johne's, and a small population had culled animals with clinical signs consistent with Johne's disease. So it is hard. There are papers out there that look at Johne's disease prevalence in other areas of the world. And the hard thing about these papers is that in most of them, we're not comparing apples to apples. So some of these papers do testing of live animals like we did, but none of them use quite the same tests as the others do. So some of them do blood testing, some of them do manure testing, some do combinations of both. Some of these tests are on animals that have been culled, and so they're taking samples from the intestines or the lymph nodes in the intestinal tracts, which is kind of the gold standard. And so you'll see different percentages in this, anything from, the single digit percentages to, higher percentages. India found 40%, Portugal was almost 25% of small ruminant herds. Canada had some high percentages, but they're all kind of looking at different populations. And so I just look at this and I say, Johne's disease is everywhere. all these countries have it. It is all over the globe. And so we knew, I mean, we were going to find it in the North Country. It was just going to be to what degree we found it. But then again, whatever degree we find it, our population that we surveyed is going to be different than any of the populations that these other papers surveyed. And so we still can't truly compare our population to other countries' populations or other research projects populations. But our goals of testing, we wanted to determine if disease was present in the herd. Certainly we could see the prevalence in the herd, or the prevalence in the animals, the subset of animals that we tested. If you're going to do testing, it might help manage or reduce or help you make decisions for how you manage the livestock that you want to raise for breeding versus ones that get sold for other purposes. If you test animals that are sick, then it'll help you manage those sick animals, or potentially make the decision to not further manage those sick animals, so that you do not spend more money and resources treating a disease that truly does not have a treatment. And then certainly if you're testing, you can use that as a tool to help reduce your risk of bringing yonis into your herd. So before I get into the tests themselves, I wanted to spend a little bit of time talking about the words that we use to describe diagnostic testing. And I kind of have this presented in more than one way because they are confusing terms that are not always easy to keep straight. And I say that given that I read and think about this on a fairly regular basis, and I still find this very confusing and frequently have to look this up and remind myself. So we're going to talk about sensitivity, which is the percentage of animals who get a positive test result who are truly positive. And so how well can the test identify the true positives? And then specificity, which is the percentage of animals who get a negative result who are truly negative. And so for lots of tests, it can be hard for the test to be good at both things. And sometimes in order to be better at identifying the positives, that test may not be as good at identifying the true negatives. So sometimes if you push a test to do better at one thing, you sacrifice the other. And this is kind of a more mathematical way of looking at it. So if you have your population of animals, and each animal is going to fall into one of these 4 boxes in the upper left. So an animal is either diseased and tests positive, so they're a true positive. They can be diseased but test negative, and that's a false negative. And when we look at that population who is truly diseased, the sensitivity are the true positives over all, that you identified with your test out of all the animals that are truly diseased. And then we have your healthy animals, and you have the healthy animals that test positive, and that's a false positive. And you have the healthy animals that test negative, and they're the true negative. And so when we think about specificity, you have your true negatives that are healthy and tested negative out of everyone who is truly healthy. So those are the kind of ways to think about it. So the testing that we have for Johne's disease don't have a great sensitivity. So we have false negatives in that population with the testing that we have. The specificity of all the tests is much better. And so you can get some false positives, but those are less common. We can feel more comfortable about the positives that we have. But for all of the tests that we have, the accuracy increases generally as the disease progresses. And so you're more likely to be able to accurately diagnose it the older an animal gets or when they are actually showing clinical signs of disease, which is really frustrating because that means that those animals may have been in your herd for a long period of time, potentially, intermittently and shedding and infecting other animals before they come up positive on a test. It also means that you've invested time and effort into this adult mature animal, and then when they're 3 or 4 or older and you get a positive test result, then it can be very frustrating. Sometimes combining tests can increase your accuracy. An animal can test positive on one but not another, so you might be able to pick up a positive earlier in the disease process if you're using more than one test. But the other thing that I want people to think about is, because all of these tests are far less than perfect, you can't easily say, oh, I got this test results when I ran this test, but I tested the same animal and I got a different test result. Because if we're doing different types of tests, so there are 3 types that we're going to talk about, or you do one type of test but on different brands, you may get different results. And that might be dependent on the day you sampled the animal, because Johne's disease can kind of change over time, and what stage of disease the animal is in. So the first test we did was an ELISA test, and an ELISA is a blood test that we do, and it's looking for serum antibodies to the Johne's disease organism. And so when you think about antibodies are what your body generates when it is exposed to a pathogen. And so over time, as the infection worsens, those antibodies levels can increase. It does mean that sometimes, the lab has to pick a point on the test where it's going to say anything above this value, we're going to call positive. Anything below it, we're going to call negative. And so certainly there are some animals that have a result that are very close to that cut point. And a year later, the level may increase to indicate to go farther above the cut point, or sometimes it might decrease and go closer, below that cut point and turn into a negative result. The hard thing is that your body is may develop antibodies at any point of exposure. So if you have an adult that then gets exposed to the organism, it can develop an antibody response. But if it wasn't exposed as a young animal, maybe it never truly develops that infection, or because it's such a slowly progressive disease, maybe that animal does not truly develop disease until beyond its natural life point. So this is by far not my favorite test, and so it is one of those things that I like to look at it and say, when I see a positive, I say, wow, okay, even if I don't know 100% that this animal is positive, this animal was probably exposed, and so I now think that there may be more somewhere else in the herd, or in a herd that animal was in at some point in its lifetime. And so especially when it's in a home-raised animal and I get more than one positive animal on an ELISA, then it makes me more concerned that there is something somewhere in that herd. But the upside of that test is that it's very inexpensive. It's easy to take a blood sample. You can test a blood sample for more than one disease. So you can send it to a lab and you can get a little bit more testing done on that one sample. And there are concerns about potential of cross reaction with other diseases. That's not super common. But it does, to me, this is kind of like the screening. This is, you're not going to necessarily be making individual animal decisions on this. This is kind of to give you a bigger view of what might be going on. You can certainly have false negatives commonly with this test. So again, not making huge decisions on this, but kind of giving you an overview. I did want to point out, so this is from a paper from 2006, and it does look at the sensitivity and specificity of this ELISA test in different species. So you can see in cattle, sensitivity on some of these, has a huge range, 15 to 87%, 55 to 98%, specificity of 55 to 98%, a sensitivity of 27 to 86. These ranges are huge. For sheep, they're, marginally maybe better. But then for goats, they're not bad on this paper. It is looking at a sensitivity, maybe in the 75 to 85% range, and a specificity, of 90 to 93%. I do also want to point out that while they're just listed for cattle, it does also look at the fecal culture, and sensitivity can be low in the fecal culture as well, but the specificity of fecal culture is very high. And so if we go back and look at specificity, that's looking at true negatives and, minimizing this false positive population. And so that makes sense, because in a culture, you're not going to have an animal that is going to have a false culture positive. The organism is either there or it's not. And so if it's there, it's, it's not going to be a false positive.
We have a question if you got a minute.
Yeah.
How this, and I don't know if we know the answer to this, but how likely are the ELISA tests to react positively to other mycoplasma, like the feces of wild birds that might be in the feed?
Yeah, so there are some papers that look at the potential for cross-reactivity with, I think it's Corynebacterium. And it really kind of goes back and forth if you look at different papers. Some papers will say that there is that potential for cross-reactivity, but it's not just that it's with a infection with exposure and immune response to a Corynebacterium. And then some that say that that's not a risk. And so it is hard to answer that question. So sorry, I didn't do a better job of answering that. But I mean, it is certainly, it's out there as a hypothesis for some. And it would be hard, because that's certainly not a disease that we're going and doing really a lot of surveillance or diagnostics for, to know how much it's truly out there to be concerned about. So is there a follow-up question for that, Betsy, or?
Nope, not at this time.
Yeah, but I do know, I would say, so the Johne's disease organism is more closely related to tuberculosis. And I do, from the work that I do for the department, I have a couple farms that when they do TB testing, they are more likely to get a suspect on a TB test, because the TB test we use for cattle is more likely to cross-react with the avian tuberculosis strain, or that is closely related to the bovis strain that we're truly concerned about. And I see that happen more in farms where they do have problems with birds in their feed alleys. And I do think that there is potentially a cross-reactivity happening on those farms, but that is that is anecdotal. And I would say that we certainly can't rule that out. I do think that that's an important reason to use more than one test, because the manure tests aren't going to have that risk at all with them. And the manure, so we have two manure tests. The first is the polymerase chain reaction, or PCR. And I think everyone's going to be a little bit more familiar with that term because of COVID testing. And PCR tests are looking for segments of DNA. And so they find a segment of DNA from the Johne's organism itself that's unique to that bacteria. And they're looking for those segments. And so you should be able to detect smaller amounts of the pathogen, because you're really looking for something very specific, and you're not trying to get it to grow or do anything more intense like we do with the culture. It just has to be there. But certainly, might not be that there's enough there to analyze. And the other so this test, we didn't get a ton of positives with this. We actually, especially for goats, did better with the fecal culture. And I do think, compared to this test in cattle, which performs very well, the pelleted manure of sheep and goats does maybe make this test a little bit more difficult to work with. And then the other downside of this is that it's just looking for that DNA, and that DNA might not be there because it's infected the animal. And we worry in cattle about pass-through infection. So if my friend in my pasture with me has Johne's and she poops and I eat a little bit of her manure, there might be Johne's organisms that enter my digestive tract, but not because I'm truly infected. And what if the test picks up the DNA from her manure that I ingested and then attributes that positive to me? So, we aren't just, there is that potential for dead bacteria to be there too, and not just bacteria that's being shed. The best option, I don't want to say best. None of these are a best option, but the kind of gold standard for live animal testing is fecal culture, because if the organism is there and it grows, we know it was there. There is no second guessing cross reactivity. When the lab grows organism in these little jars, they actually then take the organism out and run additional testing on them to ensure that it's not a contaminant of the fecal culture or a similar organism. But the downside is that it has to incubate for 56 days. And so that's almost 2 months. And it then becomes, a question of, is the information still useful 2 months later? If you end up with a positive, that animal has been positive and helping, spreading it to everyone else for, up to 2 months. So that's kind of the downside. Also, we did find, the sheep that the sheep strain is very difficult to grow, and we did not get any culture positives when we did the sheep. So testing can be really challenging. I like this paper from Perez in 1996 that actually looked at animals that were inoculated with Johne's organism and then tested at 4, 8, and 13 months after inoculation. You can see that not everyone was shedding by 4 months. Everyone was shedding at 8 months, and not everyone was shedding at 13 months. And so it does still point out that it just can be difficult to identify that organism there, even when we know that the animal has the disease, because we gave it to them. So the last test I want to touch on is a necropsy. Certainly it's less useful for, it's not what you want to do with your live animals. But I do strongly encourage anyone who has an animal that dies on the farm from unknown causes to have your veterinarian do a necropsy. It can be a very useful diagnostic test. It is certainly the gold standard for identifying Johne's disease. The digestive tract will certainly have thickening to it. You'll see changes to the lymph nodes that go along the digestive tract, and sometimes the ducts that connect the lymph nodes will be very prominent. Your veterinarian can submit samples from the digestive tract and those lymph nodes, and they can do that PCR test from the tissue itself, or they can culture the tissue itself. They'll also look at the digestive tract under the microscope through histopathology, where they actually use stain to be able to visualize the cells themselves. And they can see some very characteristic changes of the digestive tract, which will make them suspicious of Johne's disease. So getting into our results. So we had 47 people respond to our survey. 28 herds were willing to participate and had enough to kind of include. And then we had 27 that we actually did include. One did not end up working out. Myself or Dr. Ashley or Dr. Ellis, along with the herd veterinarian, visited all of those herds. We enrolled them in the Nice Gap program if they were not already enrolled. We collected blood and manure on 20, up to 20 animals. If they had fewer than 20, then we did all of the animals over 2 years of age. And then we sent our samples to the University of Wisconsin for the testing. So we had this population of 47 herds. And then we have our 27 that enrolled. Remember that we had, I think it was 1,906 goats that were part of our overall survey responders. From the 27 that we included, those farms represented 1,197 goats out of that 1,906. So that's about 60%. And we sampled 318 animals. So looking at the ELISAs, went in the wrong direction. At the ELISA results, we had 20 goats that were positive on the ELISA, or 6% of our total population, and 12 farms had those 20 goats. So that was 43% of the farms. PCR, so remember I said that we did not find many positives on the PCR. We only had 3 goats that were positive, and they were from 3 different farms. So each farm that had a PCR positive only had 1 PCR positive. And then we did the fecal culture, and we had 9 tests positive on the fecal culture, which is about 3%. And of those 9 goats, that represented 6 farms. We did have 1 goat with inconclusive results from the lab. I'm not sure. I think we're going to have to throw that goat out because that is not a test results that we can easily interpret. I know that this is a lot in this slide, but I want to kind of call your attention to a couple of things. So these are all, A, B, C, D, E. Those are each herd that participated. And I put boxes around the herds, the results for herds that had at least 1 animal as a positive, and I made all the negative bars gray. So you really just want your eye to look for the colored bars. And we did have some farms, Y, Z, A1, and G that just had 1 ELISA positive. And given the, the questions around the ELISA test, I'm willing to look at those farms and say, maybe false positives, maybe if I were to test more in those herds, maybe we would have found something. Although, you know, Z and A1 had smaller herds, so we really tested every adult we could test in those herds. So those are interesting, and I'm not as, those to me are kind of sitting in the, I'd love to retest those again next year and see how those animals look. But even with all the flaws for the Johne's disease testing, once I start to look at these herds, you know, VW, Q, P, N, where we start to see more than one animal positive, and on more than one test, then I really start to say, okay, there's probably Johne's in that herd. You know, maybe the fecal culture, you know, maybe the ELISA positive will become fecal culture positive at a later time. But once we start to see positives on more than one test, then I really start to become concerned that there's true Johne's in that herd on some level. We did have 12 farms that had at least that had one or more positive animals, so 44% of all the herds involved, and 8 farms had more than one positive animal, so a third of our farms. And we had a total of 28 goats out of our population of 318 that were positive on at least one test, so about 9% of the population. Comparing our sheep to goats, and looking kind of at the individual animals versus on the farm level, we did see on ELISA's, we did see more sheep and more of the sheep farms that tested positive using the ELISA test. For PCR, you know, this is kind of the more interesting, you know, we didn't get a lot of PCR positives in goats. We did get 6 sheep that were positive on the PCR from 5 farms. So that was a higher percentage. But the culture, you know, really seemed to perform better in the goats. We had more positives on the culture in the goats. But on a total perspective, you know, we had 9, you know, about the same percentage of sheep on an individual level. So, you know, same about 9% of our total population of sheep versus goats. But 62% of our sheep farms did have at least one animal that was positive versus 43% for goats. And again, I haven't done any statistics to compare to see if, you know, these levels of infection have any statistical significance. Certainly with our small sample sizes, you know, these aren't, you know, hundreds of farms. This is handfuls of farms. This might not be statistically significantly different. Oh, I see that there was a question. V and W only had one animal for each test. Were they the same animal? There was only, I'm going to try to go backwards. 1 minute. There was only one farm, and I could not tell you. I'm gonna guess that it's definitely either V or W, but I don't know which. There was one farm that had one animal that was positive on all 3 tests. And in both the goat population that we looked at and the sheep population, we only had one animal where they were positive on all 3 tests. And that animal was a purchased animal and had been brought in as a breeding buck. And so he was very quickly culled after those results, because every single other animal was negative. So, but that does mean that for whichever one that case was not true for, it was the other one was not all the same animal. They would have had at least 2 animals, or if not 3, yield those results. So limiting factors for our research project, certainly this was not a complete random sampling of goats in northern New York. Far from that. You know, we already are looking at a selected sample of farms that we could reach through Betsy's listserv, cooperative extension outreach, veterinarian outreach from myself. So certainly there are lots of farms that probably never even knew we were doing this. We then select within that population for herds that are willing to participate. So there's an added kind of skew to our population. And then we also didn't, randomly select from every farm that was willing to participate. We had to do, use some criteria to make our time and effort as efficient as possible with the grant funds and labor that we had to use for this project. Some herd sizes and management certainly did not allow for even our attempt at random sampling. Some of the bigger ones, we just kind of had to work with who was locked up, and if there were animals that we would have otherwise wanted to sample that were still on 100 acres of pasture, we were not going to be able to go run those animals down. And testing is not an exact diagnosis in this disease. Betsy and I are working with Clarkson University to do some additional statistics on our data sets. They're going to do Bayesian modeling, which kind of takes your results like this, where we have a data set and you can input additional information like ages and sensitivity and specificity of tests, and kind of it'll give you a range for for what the prevalence of the disease is. And so the more information that you can kind of put into it, the more accurate the range is going to be to actually have the true answer fall within that range. So we're never going to get this is the exact percentage of animals in the North Country that are infected with Johne's disease. We're going to get a range out of it to hopefully get a better idea of prevalence of the disease within our geographic region. But it's certainly here. It does seem that the farms that have it seem to have a low level of it, but that it might be present on more farms than probably think they have it. And we're going to do some additional statistics to kind of extrapolate a better prevalence range to give us a better idea. Some flocks and herds were not interested in finding out their risk. There were certainly some that politely declined being included because they, it does complicate things if they are marketing animals and then you give them a positive results and they have to kind of face that information and decide what to do with it. The hard thing is that truly kind of doing this level of sampling is cost prohibitive for most flocks and herds to do on a regular basis, to truly kind of get a better idea of their true status. I mean, if I had all the money in the world, I probably would do all 3 tests on every mature adult animal on at least an annual basis, and use that information to try to work it out of my herd. But that is cost prohibitive for most people. And so it's kind of best to, you gotta figure out what your end goal is going to be and create a balance between what is feasible to do to generate information that you can then utilize in some way. Tatiana,
so
do you want to read it?
If so let's say I want to test for Johne's so that I can tell people, maybe I can't say I'm Johne's free, but how Like if it's going to change, if some animals are going to show up maybe positive the next year, it seems like I would need to test several years in a row.
Yeah, I think that's true. I think that because we know that animals get exposed when they're young, but they might take several years to become positive on a test. And it, assuming that they're going to come positive on a test before they develop clinical disease, you're definitely going to want to commit to testing several years in a row so that you catch any young stock that were exposed to animals that were positive previous years that then themselves develop positive tests, and in anticipation that they may in the future develop clinical disease. So I don't know if anyone could say there's an exact number of years that is the benchmark. It probably depends a lot on how old animals are when you start testing. If you start with a young herd, and then follow them for a couple years, versus if you start and you had a larger range of ages. And then, if you start and you had an 8-year-old animal that tests positive, there's 6 or potentially 7 years of exposure to other, from young stock. And so even the animals that were born the previous season, those animals might need to be tested until they're 5 or 6 or 7 before you catch them become positive. And so depending on ages of positive animals that are, and when they're identified and when they enter the herd, that can certainly impact the length of time that you're kind of looking at testing.
So if you wanted to start with an ELISA test, those aren't necessarily available everywhere. And if you want, there are some other tests you can get at the same time on an ELISA that you mentioned earlier. And so you'd have to work with your veterinarian to figure out where to start the testing and with what lab. Some different of the diagnostic labs offer a little different choices of tests.
Yeah, and so most veterinarians that work with small ruminants probably have a lab that they prefer to submit samples to. But most of the veterinary labs will do Johne's disease testing, and that is the upside of doing the blood test, is that you can then also potentially test for CLA or caprine lymphadenitis or caseous lymphadenitis, excuse me, or CAE, caprine arthritis and encephalitis, neither of which are also super spectacular tests in and of themselves, but it does give you some additional information on those other diseases as well. And so that's kind of the upside of the ELISA, is that you can submit one sample to kind of get those big diseases that we worry about from a contagious perspective in goats. And so you kind of get more bang for your buck. But all the labs differ a little bit. There are different Johne's disease ELISAs out there. And so one lab is not necessarily going to be doing the same protocol as another lab. And so it does make it hard to, if you submit to one lab one year and then you submit to a different lab a different year to compare those tests, it's kind of starting to get into comparing apples and oranges a little bit. But it's definitely something to work with your veterinarian on. The ELISA is certainly the least expensive test option. And so for many herds, it's the more financially feasible place to start.
So we had another question about testing. It says, did you have any trouble getting inconclusive PCR tests because of non-specific inhibitors?
I don't
we
think
had
we
any
got any
inconclusive.
PCR results that were inconclusive. I don't think they reported that back to us for the PCR. It was just the one, there was one culture that came back inconclusive.
Mhm. And there's another question. When is there a role for an AGID or AGID blood test for yonis?
Oh, that might be beyond me. I have definitely seen it in the research as an option. I have not seen any lab offer it commercially. And I honestly don't know why.
Yeah, I haven't ever seen that either.
Yeah, I mean, I've seen it in papers predominantly, I think, out of Europe. I would have to do some more reading on and more looking into that.
So there's another question here about control measures. It says, if applicable, are there any disinfectants effective for yonis, for quarantine pens or show pens? How long is the organism effective? Sorry, I'm losing my chat here.
Oh, I
In
see
normal
it.
environments like shared pasture versus deer infection, how long to wait before rechecking?
Okay, lots of questions in this.
Yep.
Okay, so control measures. So Johne's disease will certainly live in the environment for an extended period of time. I believe that most disinfectants should be pretty effective against it, but the hard thing about thinking about it from a disinfective perspective is that many disinfectant many products, as you know, bleach is a great example, are inactivated by organic matter, which means that when you use it on things like wood or the ground, they don't work super effectively. They work really well if you have a pen that is, you know, metal panels with a concrete floor, and you clean off all the manure first, and then you use your disinfective product. It does a very good job of killing every, you know, the organism that remains. I don't know how many sheep and goat barns I've been in where disinfection is gonna, you know, 100% kill every Johne's organism that's in the wood, you know, on the wood pens. Most barns that I go into have dirt floors. And so, you know, if you have the ability to scrape a layer off the dirt floor, or, you know, if the wood in your barn is painted and sealed, disinfection is going to get you a lot further. We do certainly worry with animals out on pasture because you can't truly disinfect pasture. You certainly might get enough kill during, you know, our winter season that, you know, the next season there would be less organism load there. How long is the organism effective in normal environments? Shared pasture. So certainly it's gonna be there through the summer season.
And
The
what
winter
about
should not.
spreading
Mhm.
manure? I mean, that's one way that it gets around in the pasture if you're spreading it on the
Yeah,
crops or the pasture.
yeah, and so we certainly do, you know, the dairy industry and thinks about that when they spread manure on fields and stuff like that. Anything that you can do, you know, if you're worried about a field, if you go through and brush hog it to get some of the volume off of it, so more air and sunlight can dry out the soil, that might be helpful at knocking back the levels. But there's so many variables in there, it's hard to give an exact answer. But I do like that this person asked about deer infection, because deer can get Johne's disease too. I don't know if God, that'd be an interesting thing to look up. I don't know if anyone has done any research on Johne's disease in the wild deer population. There certainly is not a commercial Johne's disease test for deer. So I'm going to say that's probably, there's not a lot of information out there. You know, I don't know how many deer are living long enough to truly develop Johne's disease. I bet that's very dependent on the region of the country you're in, and how long to wait before rechecking. I think that how long you wait before you recheck is going to be really dependent on how you're using the results, and what management decisions you're making and what your goals are. And so I think your duration of before you recheck should be something that you work with your veterinarian on. Yeah, I think that's a individual one-on-one conversation with your veterinarian to make that decision.
So we have another question about what about Lyme? I would
I'd have
think that'd
to-
be the same as your disinfectants, pretty much.
Yeah. I mean, I think that if you had a dirt floor barn, spreading Lyme down on it might help knock it back. It certainly does help reduce bacterial populations. Most of my knowledge about Lyme as a disinfectant comes from my days doing milk quality and mastitis work with Cornell. And we did see that when Lyme was added to bedding, it did a very good job of controlling bacterial populations in bedding, but only for about 48 hours, you know, but we did look at it a lot more with manure solids. And so you're really taking a bacteria heavy substrate and trying to keep it in check. But even when you look at sand bedding, you know, once you start adding organic matter to it, the efficacy of Lyme at controlling bacterial populations goes away pretty fast.
So Janet's asking us, does it survive on hay harvested from fields spread with infected manure?
I would be far less concerned with dry hay than
Haylage or
haylage.
valage?
Yeah. And certainly even with haylage, it's going to depend on, you know, the process for ensiling. And I'm sure that, you know, inoculants can impact it too, with the good bacteria might be able to keep some of the Johne's in check, but that's probably something there is more research on the dairy side for that I have not looked at, because I'm not generally a crop person.
So we have another question here. What is the age window that animals can become infected with Johne's? Not sure we know that exactly, but.
Yeah, certainly our young stock are going to be our most at risk. But infection is very dependent on pathogen load along with age of exposure. And so, you know, if you think about it and you have a very young animal, that animal probably does not need nearly as much pathogen exposure to then be at risk for developing infection later on, versus if you have a slightly older animal that gets a very large load, that animal may still also develop infection later on. It also, you know, age and pathogen load will also potentially impact age of onset of clinical signs. And so the earlier it is, or the bigger the load is, you know, the potential earlier they might start showing signs of disease. But I will say that for farms that are trying to manage a population with potentially infected animals, I'm far less stressed out if we're going to take, if you have a 2-year-old animal or a 3-year-old animal that is otherwise negative, and putting that adult animal with a positive, because that animal is mature enough, has a mature enough immune system, is far enough away from that young stock age range, that I would be less stressed out exposing adult animals to known positives, if you're trying to manage two populations.
So here's another thing we talked about the other night is, was there a high degree of interaction with cattle, milk or manure in herds that had positives? And we sort of said there'll be another good research project.
Yeah, and actually we can go back and we do have some of that information, and it would be easy for me to spend 15 or 20 minutes looking at and seeing, that those 27 herds, out of those, the 12, whatever percentage, the 40 whatever percentage that were positive, how many of those also had cattle, or I would include sheep in that, on the premise as well. We did not ask, and I don't know what potential interactions the goats have with bovine milk, and that was not something we asked, but we could certainly try to look at who else had other ruminant species and see if that correlated with herds that were positive.
Yeah, and we should point out that it's not just dairy cows that get yonis, but also beef cattle. So if you're multi-species grazing, it's something to consider at least. I think there's another question here. At what age can animals be reliably tested?
So that's a good one. You guys probably noticed that for our research project, we only included animals over the age of 2. And so I guess that depends on the hard thing is what's the definition of reliably tested? So this is the hard thing about this disease. You can test an animal at 6 and she might still become positive at 8. I don't think there is any point in testing animals younger than 2, unless you already know that Yonis is in the herd to a very significant degree, because the chances of, it's such a slowly progressive disease, the chances of you finding positives in younger stock is going to be low. And so, unless I tested a herd or a flock and found a huge number of positives, I wouldn't test animals younger than that, unless I had younger animals showing clinical signs consistent with disease.
Okay, that seems to be the last chat question. Hopefully we haven't made you all depressed about your potential yonis in your herd. It's just something you definitely want to be aware of and try to avoid if you can. I'm just giving another minute or 2 here in case people think of other questions. And I think you could also email me and I can pass it on to Jess, or you can email Jess if you have her email, and we can try to answer any other questions you have as well.
Yeah, and we, certainly we really appreciated the herds that participated in this project and the flocks that participated when we did this in sheep. Even those that didn't want to be sampled, that just answered our questions, that was really helpful. And then we had, a really strong group of veterinarians that were really supportive of this project. They helped with sampling, they helped recruit farms, they helped schedule. They were really instrumental in getting this project done.
Right, and they had to do the follow-up for people too with their results. So
yeah, they got to answer all the hard questions.
So we have a whole lot of ideas for more research projects related to yonis. As you can see, there's not answers to all the questions that we'd like to have. So hopefully someday we will get the answers to those. Maybe not we, but somebody. Great. Well, thank you all for joining us tonight. This will be recorded. It is being recorded, and we will send you a link to the recording. And we have another chat here. Hold on. think I'm having a little trouble getting to it. Sorry.
Oh, it's Janet saying thank you.
Oh, here's one. I guess that's just to me from Tatiana. We can talk about that one later. Yes, and lots of thank yous. Well, we're glad you could join us and that we could share this information with y'all.
Yeah,
It was a
thank
really
you all
interesting
for
project to
listening.
work on. Yes. And it's really nice to have a bunch of veterinarians that are interested in sheep and goats. So that was also really nice to see in our area. Well, everybody have a great night. Thank you for joining us. And we'll probably have some more sheep and goat webinars coming up this winter. I'm in the process of trying to set one up on solar grazing, interviewing somebody I know that's a solar grazer, and possibly some others, because we're certainly getting a lot of phone calls about that at the moment. A lot of people are being approached. So we do have some good information on what working with solar leasing. You know, if you're going to do a lease, some things to watch out for, some good webinars on those. So if you have questions, feel free to give me an email and I'll send some of that on to you. So great. Thank you all for coming and have a great evening.